Quand tu veux, plutôt tu peux tricher.
Bonjour à tous. Welcome everyone. So sorry for the delay. We had some technical issues and we got a bit late today, but yeah, thank you for joining. So it's my great pleasure to introduce Marie Otomwa from Kenya, from the Kenyan Marine and Fisheries Research Institute. So she's been working in fisheries for over more than 10 years. And she'll be presenting the main institute, KMFRI, but also a research. And I'm trying to set up a strong collaboration with KMFRI, with different people, including Marie. So hopefully it's not the first time she'll be visiting us. So thank you so much for your time again. And yeah, the floor is yours.
Thank you. Hello, everyone. As you've heard, my name is Mary Ontomwa. I work with Kenya Marine and Fisheries Research Institute. I hold a BSc in Fisheries, Masters in Fisheries, and also I recently completed my PhD in Fisheries from the University of Pwani University. And I do associate myself with WIOMSA and the Women in Marine Science, and also the Western Indian Ocean Career Scientist Network. So I'm today presenting to you about Kenya Marine and Fisheries Research Institute and part of my research work which I do in the institute.
Yes.
So our institution is a national institution which was initiated in the 1979 under Science and Technology Act, CAP, but now it was repealed by the Science and Technology and Innovation, and it was changed to be a research scientist by the year 2013. Our mandate is to undertake research in marine and freshwater systems, and also we do aquaculture, and we do various activities like chemical and physiochemical oceanography in order to provide scientific data and also information for the sustainable development of the fisheries. The vision is to be an excellent center in research that's for both marine and freshwater systems, and also the mission is to generate and disseminate scientific information for the sustainable development of the fisheries. And our core values are integrity, transparency, and accountability, professionalism, teamwork, and also equity and equality. So our institution is not only a one-center institution, but we have different centers that are located in the entire country. So we have centers, Mombasa is the headquarters. We have Kisumu Center, we have Sagana and Kegati. These two centers, they deal with aquaculture. And we also have smaller institutions that are stations. We have Naivasha, Baringo, Turkana, Sangoro, Gazi, and the Nairobi Center. Nairobi Center is a liaison office which tries to liaise between all these other centers. As you can see here, the centers are distributed in the entire country. And there are more centers that are upcoming, but they have not yet been fully established. If you want to know more about KMFRI, you can use the net there. That is www.kmfri.go.ke. If you go there, you will see more information about KMFRI and what we do in the institution. So in KMFRI, we have different directorates. We have 4 directorates. We have the Directorate of Ocean and Coastal Systems and Blue Economy and Research. That's where our marine coastal fisheries lie. We also have the Directorate of Fresh Water Systems Research. We have the Directorate of Aquaculture Research and the Directorate of Socioeconomics Research, which tries to link what we do with the communities. And our main research areas is fish stock assessment. We do plastic pollution, we do cage culture, fisheries biology, post-harvest losses and value addition, and also we do community involvement. Our institution has about 122 research scientists, and apart from this, we also usually receive interns, and also we do receive student attaches from universities who work with us for a short period, depending on the contracts signed. So we have different departments under the various directorates that I've mentioned. We have the Oceanography and Hydrography Department. Also deals with it's also called the Limnology Department. We also have the Marine and Coastal Fisheries Research Department. Sorry for that. We have the Aquaculture Department and Socioeconomics Department. All these departments do the work which I've mentioned earlier. And above all, we also have the Information, Communication and Technology Department, which tries to capture the data which we collect and archive it for in case there's need for reuse, or in case someone wants to request on it, they can get it through the department. So KMFRI does not work in isolation, but we also partner with different organizations. We will partner with our government of Kenya, the other different organizations, Kenya Wildlife Service, WIOMSA, non-governmental organizations, UNDP, and also World Bank, and also the Food and Agricultural Organization, that's FAO. And through this partnering, we have done so many projects and with the different organizations. And currently, we're running a project on KMFRI, that's the Kenya Marine Fisheries Socioeconomic Project, which is running under and it's being funded by World Bank. So the challenges that we encounter in our institution is that sometimes we have research underfunding in all departments, all directorates. We also have limited capacity in assessing the effects of climate change on the fisheries, that's in terms of diversity and stocks. And sometimes we have limited capacity in assessing transboundary fish stocks, especially for the pelagic fish. And there are limited studies on the contribution of marine fish to food security and the nutrition. We had one student who did his PhD in Lakansida, that is Johnson Omkoto. He was able to do that study and was able to finish his PhD on that. So those are some of the challenges which we encounter as an institution. So that's all about our institution. I'm now going to move to the work that I do as a research scientist at KMFRI. And I'm not going to present all the work, but I'm going to present part of the work which I did during my MSc. And also I'll present part of the work which I did during my PhD, not all of it, but one objective. So during my master's, I did determine hook size selectivity for the handline fishery along the south coast of Kenya. And as an introduction, we understand that the small-scale fisheries are important in supporting livelihoods, and also they provide food security and nutrition for the coastal communities. And annually, by that time, when I was doing my MSc in 2017, the landings for the 2016 year were about 9,000 metric tons per year, which are valued at about 1.3 billion Kenyan shillings. And these fisheries support about 13,000 fishers. And the small-scale fisheries are exploited using various gears. For example, we have the pitch seines, we have the basket traps, we have the hand lines, the spear guns, the reef seines, among many others. So my interest was based on the hand lines, because we find that the hand lines had been increasing over the years, and this was led by the increase in fishing effort. In the south coast where I did my study, I found that about 50% of the hand lines are used in the south coast, which accounts about 1,800 hand lines. So that's why I concentrated my study at the south coast. And when we talk of gear selectivity, it's a measure of how different gears select fish of different sizes in a fishery. And in this case, gear selectivity is important because it helps us to set capture limits for a fishery. And it also tries to illustrate the impact of the fishing gears on fish stocks. So for sustainability, we need to understand the gears, how they select fish and what impact they have. That's why I had to do the selectivity of the hooks that are used in the handline fishery to provide information on the fishery. That is important in the management and regulation of the fishery. So the main objectives of my study were to identify the species and the sizes of the fish which are caught by the different fishing hooks that are used in the handline fishery, and also to determine the size composition of the fish species caught by the different hooks selected during the different seasons and also at different places. The study, I did it at 4 sites. I did it at Wasini, the shaded regions on the map, and also I did it at Mpunguti, Nyuli, and the Waga. Wasini is also called Mundini. So those are the study sites which I selected from the study because they showed the handlines were rampantly used in those areas. That's why I selected those sites. And the description of the study area is as illustrated there. So during the survey, I did the survey in 2 days in a month for the different 4 sites. And I did my study from January to June 2016. And I used 5 different hook sizes, hook number 8, hook number 9, 10, 15, and 16. The numbering of hooks is given in a way in that the largest hook has the smallest number. So that's how the hooks are numbered. So when we talk of number 18 is a small hook, hook number 16 is a small hook, and hook number 8 is the largest hook. And during the setting of the experiment, we could set 2 hooks on each of the handlines. And after that, we could attach a metallic lead, that's a sinker, so as to allow the lines go deep at the bottom to capture fish. And after capturing fish, we could preserve the fish in the boxes and then transport them to the landing sites, where we could identify the fish using various identification fish guides. And after identifying the fish, we measured the sizes, that's both in total length and also the body weight of the fish. So to assess selectivity, I had to cluster the length sizes of the fish which were caught by the different hooks into 2 centimeter length classes. And I had to adopt Holt's 1960 model to determine the optimal size lengths that are caught by the different hooks using the formula. That's formula 1 to 6 as illustrated on the board. You can see what C1 and C2 implies, and also SF is the selectivity factor, and the optimal length is represented by LM1. So the others are clearly shown as it is illustrated by the formula. So further, I use multivariate ANOVA to assess differences in the mean length of the fish caught by the different hook sizes among sites. And also I did K-dominance analysis to assess species dominance among the stat sites and for the different hooks and also by season. And also we did hierarchical cluster analysis to determine the similarity of the species which were caught by the different hooks. So the results of my study show during the survey period, I was able to capture or to catch 966 fish. And as you can see, most of the fish were caught from newly sighted. And the distribution of the fish in terms of numbers is shown on the bar chart. So in terms of species abundance, these are the species which were caught during both the southeast monsoon season and also during the northeast monsoon season. They were not quite different in terms of numbers, but the most dominant species which was caught during the same season is the Letrinas bolbonicus, and during the northeast monsoon season was Letrinas lenchan. So when it comes to the sizes of fish caught by the various hooks, I did the mean sizes by the different hooks. That's the general mean sizes. We find that usually we recommend size length of 24 centimeters. That's the table size of fish that has to be caught during the fishing. But we find that hook numbers 16 and 15 were caught fish with the mean sizes which were lower than the hook numbers of hook numbers 10 to 8, which means that the larger the hook, the smaller the size of the fish which is caught, and that's in terms of the number of the hook. And also, the smaller the number of the hook, the larger the size of the fish which is caught. So in this case, large-sized hooks caught large fish, and small-sized hooks caught small-sized fish. So when we compare the selectivity, we find that the mean sizes of the fish caught by the hook numbers 10 and 9 were not significantly different, and also those ones which were caught by hook numbers 16 and 15 were also not significantly different. Similarly, for hook sizes number 8 and 10. So when it comes to selectivity, I did selectivity for the most dominant species, that is for the Lucianus fluflama, Letrinas lenchan, and Aplon fersens, Letrinas bacletas, and Letrinas bolbonicus. So from the modeling using Holt's 1993 model, we find that hook number 16, 15 caught a small-sized fish for the case of the 4 species apart from Letrinas bolbonicus, and also hook number 9, 8 caught large-sized fish, although we find that the 2 species, that's Lucianus fluflama and Aplon fersens, were not caught by hook numbers 10, 9, and 8. So assessing the sizes of the fish which were caught by the various gears, we find that when I looked at the length at first maturity compared from that's adopted from fish base, we find that all the hooks that were used captured mature Letrinas bolbonicus. That is the fish which were the optimal length is for all the hooks is above the length at first maturity for the species. For Letrinas lenchan, we find that hook number 15 caught small-sized fish. That is the optimal length of the fish which were caught were immature. And also for Letrinas rubidopaclatus, we find that the hook sizes number 8 and 9 caught immature individuals. So for latrinus lentian also, we find that the hooks are caught relatively immature fish, and also for aprion fersens. So when it comes to dominance of the species which were caught during the study period, we find that high dominance of species were caught during the northeast monsoon season, and also the high dominance of species were caught by hook sizes number 15 and the number 16, which were the small-sized hooks. So it indicates that the small-sized hooks caught many species compared to the large-sized hooks. And from the sides, we find that the high dominance of fish were caught at Mpunguti. And this could be because Mpunguti is closer to the MPA, that is the Kisite Mpunguti Marine National Reserve, which is there could be a spillover of fish from the reserve. That's why maybe we got a high dominance of species at that particular site. So the similarity, we find that hooks number 8 and 9 caught similar species, and also hooks number 10 and 15 had similar species composition during the study period, as shown by the dominance analysis, the hierarchical analysis. Now, for discussion, we find that high abundance of species diversity occurred during the same during the NEM than the same due to migration of fish to deeper thermocline waters, which occurs during the southeast monsoon season. And also the small-sized hooks catch high numbers of fish, while large-sized hooks catch less numbers. That's in terms of abundance. And the large-sized hooks capture more small and immature fish, whereas large-sized hooks capture more large and mature fish of longer lengths, resulting to high catches. So in conclusion, when we vary the hook sizes in the handline fishery, we get significant abundance variations, and also variations in the sizes of the fish that are caught. And also the selectivity of the different hook sizes used in the handline fishery also differs by species. And if we increase the sizes of the hooks, it can lead to the capture of mature fish. And therefore, we recommended that during the handline fishery, there is need to use the large-sized hooks so as to conserve all for the sustainability of the fishery. Although, after disseminating these findings, we do not implement the recommendations because KMFRI is a research institute and it's not mandated to enforce the regulations. So we just recommend to the State Department of Fisheries to undertake the enforcement. So that's the end of that part, and I acknowledge my supervisors, Professor Flanda of One University and Dr. Kimani from KMFRI, the fishermen, and also the field technician, and also appreciate WIOMSA and NACOSTI for funding the project. So I'm now moving to the second part of my presentation. That is a part of my PhD work, that's biomass estimation and the stock assessment of pelagic fish of the Kenya marine waters. So as we understand, the marine pelagic community is the largest rim, covering about 1,300,000 meters, which is equivalent to 71% of the Earth's surface in volume. And we find that the Indian Ocean covers about 45% of the pelagic marine fish landings. that they provide about 45% of the marine fish landings. And Kenya, as a country, we produce about 20% of the marine fish catches. So these pelagic fisheries, they are important because they supply 80% of fish food for human consumption. And also, we find that these pelagic fisheries in Kenya, they provide nutrition, they also provide finance and economic support to both the industrial and the artisanal fishers. In the 70% of the coastal population rely on the marine capture fisheries for about 80% of their income. So these pelagic fisheries, they are exploited by industrial fishers who are majorly exploit offshore waters. And these offshore waters are about 20 to 200 nautical miles, and they target the large pelagic fish, which forms about 37% of the treated fish. The small-scale fishers, they only do fishing at the inshore waters, which is within the 2.5 to 3 nautical miles, and they target the small pelagics and also the medium pelagic fish. Apart from the industrial and the small-scale fishers, we also have recreational fishers or the sport fishers who also do fishing, and they target the medium pelagic and the small pelagic fish. So regardless of the contribution of this pelagic fish, information for the offshore pelagic fish stocks is limited. And it is because of lack of data or poor data records and lack of reporting of catches, and also limited resources to study the extensive EEZ. So that's why we try to conduct this study to generate information which is useful for the management and regulation of the EEZ fisheries. So the objectives of the study one was to evaluate the Kenya longline pelagic fishery and also to describe assemblages of the pelagic longline catches. And also on to the right-hand side, there are specific objectives for each of the objectives. For purposes and for the interest of time, I'm only going to present results on the second objective, that is the assemblages of the pelagic longline fish catches. So for the longline pelagic fishery assemblage characters, I did a survey in the EEZ as shown in the map. And I did in 3 sections. That's the nearshore zone, zone 1, zone 2, and zone 3. And sampling was done at the different parts of the EEZ as depicted on the map. And we conducted 5 surveys lasting for 20 to 24 days each using a vessel that is a fishing vessel SIMA 2, which we leased during the study period. We did set the long lines in the evening, and hauling was done in the morning. And the survey strategy is as shown in the lower figure 2. So during the experiments, we used frozen shark mackerel as our bait. So after landing, we could identify the fish and measure the different lengths. We used different mensuration, total length, focal length, fork length with disc length, and the lower jaw fork length, depending on the species which was landed. And the data which was recorded is shown. You had the number of hooks that were set and retrieved, the location of fishing in terms of coordinates, the start time and the end time, and also during the survey, we could record the fish which were discarded. So I described the catch per unit effort using that formula. That's the total number of fish divided by the total number of hooks which were deployed times 1,000 hooks, which is the standard number of hooks that is used. And in this case, I used numbers to determine catch per unit effort, because the variation of weight for the different species may conceal the weight of the smaller sized fish. So in this case, when we try to do assemblages, we determine the numbers, how much in terms of numbers are given in a given locality. That's why I used catch per unit effort in terms of numbers and not in terms of biomass. So we also did determine diversity using these indices. And we did statistical analysis using the methods that are being listed on the lower part of the slide. So we managed to do 110 sets, and we managed to deploy that 6,000 hooks during the survey period. However, at the 3 different zones, there was some slight variation of effort in terms of the number of hooks which were deployed. In total, we managed to get about 4,200 fish species, fish specimen belonging to 50 species. And this belonged to 34 genera and 23 families. So this composition is quite different from the first objective. When we did species composition for the logbook data, we found that it's only 20 species that were recorded in the logbook data, which implies that there's some bit of misreporting. So the dominant species we had were swordfish, we had blue shark, and also big-eyed tuna as the dominant species. And some of the species that occurred rarely, we have the albacore, we also have wahoo, and we had the great white shark and the smooth hammered head shark. So when it comes to variation of catch per unit effort in space, we had for swordfish and yellowfin tuna and also silk sharks, that's Cacharinas falciformis, their catch rates decreased from the nearshore zone to the offshore zone. But for the blue shark, that's Prionase grauca, the catch rates in terms of numbers increased from the nearshore zone to the offshore zone. For bigger-eyed tuna, the catch rates were highest at the mid-EEZ zone. And for the Isurus oxynicus and the Galliasado cuvia, we had, plus the common dolphin fish, we had their catch rates high at the nearshore zone, while for the Cacharinas longmanus and the other 3 species, here they had high catch rates at the offshore zone. So when it comes to monthly catch rates, we find that the mean catch rates were highest during the month of July, with the nearshore zone having the highest catch rates and the offshore zone having the lowest catch rates. And also during September, that's when we had low catch rates. However, the mean catch rates, that's CPU, was not statistically different among the various months of sampling. So when it comes to species by month, we find that swordfish, that is Cephalus gladius, had high catch rates during the months of May and July, while blue shark had high catch rates during the month of July, and the Cacharinas longmanus had high catch rates during the month of June, while tunus opesas, that is the bigger tuna, had high catch rates in May, and the tunus opesas had high catch rates during the month of June. So when it comes to species diversity, we find that there was high species diversity at zone 1, and also we had high dominance of species at zone 1 at the same time. And the dominance was also highest in the month of August compared to the other months. So when I did the biology of the species, we found that for swordfish, the L50 is 127.7 centimeters. And we find that at the different zones of fishing, that is zone 1, 2, and 3, about 50% of the fish which were caught were mature. And also, most of the mature fish were caught during the month of July. So for the blue shark tuna, that's pronounced grauka, the length at the L50 was 271.9 centimeters. And also about 40% of the fish which were caught at the 3 different zones were mature, while in the month of October, we find that almost all the fish were caught during the month of October were immature. And very little, very few species, not species, but numbers of mature fish were caught during the month of September. Sorry. So for yellowfin tuna, the L50 was 146.4 centimeters. And zone 2 had the highest proportion of mature individuals, and while high proportions of mature individuals were observed during the months of April, May, and July. And for the big eye tuna, the L50 was 158.3 centimeters. And also, we find that a large proportion of mature, the proportion of mature fish increased from the near zone, that's zone 1 through zone 3, and the proportions of mature individuals was recorded during the month of April, May, and September. So from this, we find that catch rates vary by time, season, and depth, and it's influenced by the amount of effort deployed during fishing. And also, we find that there were some high levels of discards that were observed during the longline survey. About 0.1% of the fish were discarded. And therefore, the recommendation is that the authorities should deploy scientific observers in the longline fishing vessels to ensure comprehensive recording of discards to guide management. Although there are scientific observers on board, but not in all longline fishing vessels. And also, during the survey, we encountered with the ETPs, that is the endangered, threatened, and protected species. We recommend management to monitor and control the capture of these species. And the near zone exhibited high species assemblages and high species richness compared to the other zones. Therefore, we recommend that this prioritization of the conservation efforts in the near zone, and also most of the individuals of yellowfin tuna and Bigger tuna captured at the near zones were immature. Therefore, we recommend management to initiate restrictions of tuna fishing at the near zone to protect and conserve immature individuals. So I acknowledge my supervisors. I also acknowledge KMFRI scientists for cooperating during the survey period, the vessel captains, and also I acknowledge the government for funding this work. And I acknowledge One University and also the Kenya Fisheries Service for providing logbook data for the other objective, and the PESA company for providing me with the vessel which I used during the survey. Asante sana. Thank you.